Hello, and thank you for joining Cure Alzheimer's Fund today for this important presentation. I am Meg Smith, CEO of our organization, and I hope you will allow me just a few minutes to introduce CureAlz. We were founded with one mission: to fund research that will accelerate prevention, treatment, and cure for this disease. To that end, since 2004, we have deployed more than $200 million toward extraordinary research pursued by brilliant and determined scientists around the world. Our dedicated donors make all of this possible, people who share our drive and commitment to end this disease and its terrible impact on patients, families, and our communities. We are grateful for their trust in us, and we are determined to honor it by finding and funding the vital research that is the only way to achieve progress. Our board of directors and trustees hold us accountable to our mission. They and certain other supporters cover all of our operational overhead. This generosity and vision mean that our other wonderful donors know that 100% of their gift goes directly to our research program. At CureAlz, our only goal is to find solutions to help the Alzheimer's community. We do not pursue profits or financial returns. All of our scientific decisions are made by active researchers who themselves are at the forefront of discovery. Our research leadership group guides our scientific decision-making. They are experts in scientific specialties relevant to Alzheimer's who lead labs at top academic research institutions around the globe. They determine what investigations we will support, always ensuring we pursue the ideas that science and data tell us are the most likely to make a real difference for current and potential Alzheimer's patients. Thank you again for joining us to hear about the science and progress that matter in Alzheimer's. We hope you will continue to join us in our mission to end this disease.
So I guess a little about myself I could start out with. I got my MD PhD at Vanderbilt and my neurology training at University of Pennsylvania. I see patients 1 day a week. My fellowship training is in dementia, so I work in an Alzheimer's clinic. And then the rest of the time I run my lab, which is a mix of basic science and kind of translational science, but mostly cells and mice and that sort of work. We do a little bit of human studies. So it's kind of a mix. And certainly, I think early in my career, Cure Alzheimer's Fund is really critical in helping me get things off the ground. I mean, obviously, when you're starting out, it's more difficult. You have ideas, but not necessarily a reputation or a lot of data to support them. And so Cure Alzheimer's Fund is so important. I'm going to move on and just talk a bit about Cure Alzheimer's Fund. So in 2018, I got my first funding from Cure Alzheimer's Fund to study these reverb alpha proteins, which are circadian clock proteins. I'll point out some of that research as we move along. And then I recently got a second round of funding from them to study a different topic, which is jet lag and how that affects Alzheimer's disease. I'll discuss that data today as well. I think this illustrates something that I thought was amazing in that Cure Alzheimer's Fund actually came back to me to see if I had any new interesting ideas to potentially be funded. I thought I got one thing in 2018 and that was it, but they kind of keep a relationship, and I feel like that's a really unique phenomenon that a lot of other funding agencies don't do. You're kind of one and done. But the Reverb Alpha project from 2018, the data that we developed from that led to several publications, as well as NIH grant and NIH R01 grant, which I'm in the 4th year of now, planning to renew. Actually just submitted the renewal for that. So that's kind of become a well-funded project that I hope you think is interesting. I think it's yielded some interesting things, some potential. And I think these Cure Alzheimer's Fund grants are so critical for a number of reasons. I mean, the previous slide, I didn't see that before I made my list, but the flexibility and the rapid nature, getting grants from the NIH takes a long time. You have to basically have the project done before you get a grant from the NIH. And so if you have an interesting idea that you need to get going, Cure Alzheimer's Fund has the flexibility to sort of fund that in its early stages, rather than waiting years for the NIH to sort of come around, which they eventually do, as evidence from the grant I had. Obviously, funding earlier stage projects that have interesting ideas and maybe novel hypotheses that the NIH might balk at first until you have a boatload of data to support it. And I think this being connected with an amazing group of scientists. So the scientists that are funded by Cure Alzheimer's Fund are really amazing. I mean, it's an incredible group of people. And when you get to go to these meetings and interact with these people and collaborate with them, it really creates this network of people that's hard to come by otherwise. So it's really a one-of-a-kind organization. I'm really grateful to Cure Alzheimer's Fund for helping me out. So let me tell you a little bit about what I've been doing and about this field, why study sleep and circadian rhythms and Alzheimer's disease of all things. But I have to start out with a little bit of background on Alzheimer's disease, which this may be review for many of you, but there's obviously two pathologies in the brain in Alzheimer's disease. You've got these amyloid plaques, which are extracellular conglomerations of this A beta protein, among other things. And then you have the tangles, neurofibrillary tangles, which are aggregated tau protein inside neurons. And we know now that the amyloid plaques are the first thing to show up, at least the first thing we know about at this point, and that they precede and probably provoke tau. So we think that amyloid plaques somehow induce this tau pathology. And the tau pathology shows up later, but all this happens before symptoms. So in this graph here, symptoms are starting to show up where this line is. That's when people are starting to have memory issues. And obviously amyloid is there probably 10 to 20 years before that. And tau is probably there 5 years before that. So there's this huge period of time, this preclinical phase during which people have pathology in their brain, but have not yet developed the symptomatic aspects of the disease. And so this is a really unique opportunity, I think, that because we have biomarkers to detect these things to potentially slow the course of the disease or even prevent the symptomatic phase of the disease, which is exciting. The question we have is, where does sleep and circadian rhythms kind of fit into this? So we know that people that have amyloid plaques in their brain have sleep and circadian abnormalities even before they develop memory problems. And I'll show you some data to support that. But what we don't know is it happening even earlier than that? People in their middle age, if they're having poor sleep or poor circadian rhythms, are they at higher risk for Alzheimer's disease? And so I'll talk about that to some degree as well. I don't think we have a firm answer on that yet. So we know that sleep is disrupted in patients that have Alzheimer's disease that are symptomatic, that are developing dementia. Patients have poor sleep efficiency. They're in bed a lot, tossing and turning. They don't sleep as soundly as they should. They nap more during the day, probably because they're sleepier from not sleeping well at night. If you put an EEG on their head and look at the brain waves, there's abnormalities in the brain waves. They have less of this really deep, slow wave, non-REM sleep. They have decreased of the dream-related REM sleep, rapid eye movement sleep. But the main thing that we see is this fragmentation of the sleep-wake cycle. And that's where people are waking up more at night than they should, and they're sleeping more during the day than they should. And so the difference between day and night becomes less. I think there may be a circadian underpinning to this, but we'll talk about that. And we know that these fragmented sleep-wake cycles and increasing naps are apparent before in people that have amyloid plaques in their brain and have biomarkers that are positive for plaques before the onset of actual memory loss. So we think if you could intervene on these things, you could potentially maybe stave off the symptomatic component. One of the big questions I always get is, well, if I don't sleep enough, am I going to get Alzheimer's disease? How much do I need to sleep? Do I need to sleep 10 hours a day or 3 hours a day or whatever? There are some studies of this. They're not great, but they're out there. So it suggests that I kind of made this summary graph here that if you're getting between 6 and 8 hours of sleep, it doesn't have a big impact on your risk for Alzheimer's disease. So that's most people, Now that is self-reported sleep. So all these studies, they basically just ask people, hey, how much do you sleep? They're not really looking at EEG. They're not looking at objective measurements of sleep. They're looking at asking them questions, because a lot of times these are done after the fact. You ask people when they're 80 years old, oh, how much did you sleep when you were 50? They may not be very accurate. So that's a big problem with these studies. But the data does suggest people that report not sleeping a whole lot, like less than 5 hours a night, do have a higher risk of Alzheimer's disease. And there's multiple studies showing that. And probably the best study is this one that was published a couple of years ago in 2021, where they used this data from this thing called the Whitehall study, which is kind of like a big epidemiologic study in the UK, or they follow people for like 50 years and they fill out little questionnaires every year and they have some measurements of different things. And essentially what they found is when they talked to people when they were 50 years old, if they reported getting less than 6 hours of sleep, that 25 years later, their risk of Alzheimer's disease was about, or of dementia, not necessarily Alzheimer's disease, of some kind of dementia was about 28% higher. So 1.28 times the normal rate. So if you were, in middle age complaining and not getting enough sleep, it does put you at higher risk. Now, we don't know how much sleep these people were actually getting. People, this is what they report. It gets even worse at age 60. People that were reporting less than 6 hours of sleep a night, they have about a 50% increased risk of developing dementia 25 years later, 20 something years later. So it suggests that this middle age sleep is important, but we need better studies, to be honest, where you can measure people's sleep when they're younger and see if that portends, risk later in life. Because there's this long prodrome to the disease. As I mentioned, it takes 15 to 20 years for these amyloid plaques to build up in your brain and actually seem to cause memory loss. So what is the evidence kind of mechanistically linking sleep to Alzheimer's disease? Why would we think these two things are connected? I think the first big discovery was from my mentor, Dave Holtzman, here at WashU. He published in Science in 2009. He found that when we looked at amyloid beta protein in the brain, so you can measure amyloid beta in the fluid of the brain by putting a little probe into the brain of a mouse at every hour over the course of 2 or 3 days. And what they found is that the levels of this amyloid beta go up and down with a 24-hour period. So a diurnal pattern of A beta levels. And when the mouse is sleeping, so the mice sleep during the day when the lights are on, their amyloid beta levels go down. And when they're awake and running around and having a lot of brain activity, their amyloid beta levels go up. And actually, this is pretty well explained by neuronal activity. So when your brain is really active, you produce A beta. That's been shown by several groups, including Holtzman's group. So the idea is when we're sleeping, our brain is quieter, it's calmer, it's not making as much A beta. And then when we're awake, we're making a lot. And that if you're constantly awake, then maybe you're constantly having higher levels of A beta that end up aggregating into plaques eventually. And so they showed that by taking mice. These are all transgenic mice that express a human gene, so they get amyloid plaques. And they sleep deprive them. So they put them on these little platforms over a water container so they can't sleep or they'll fall off the platform. And when they do that, the mice, the black lines here show the mice that were sleep restricted, they get more amyloid plaques, sort of in all different areas of the brain. You can see the picture here. This is a sleep deprived mouse. So sleep deprivation seems to accelerate amyloid plaque formation. And that was kind of the first scientific link between sleep and Alzheimer's disease. And there's been more since then. I think I took the picture out, but Holtzman's lab actually has another paper showing that tau has a very similar sort of thing. That the levels of tau will go up if you sleep deprive a mouse. So both amyloid and tau seem to be sleep dependent to some degree. So mice are fine, but what about humans? So one of my other colleagues here at WashU, Brendan Lucy, does these really demanding experiments where they put catheters into people's spinal canals and drain fluid out of them, and they sample their spinal fluid every hour over a day. And during that time, they either sleep deprive people or they let them sleep normally or they force them to sleep with a drug called sodium oxybate, which is used for narcolepsy. And they measure amyloid beta. And what they basically found, looking at these graphs is normally when you're a person, the blue line is a normal control people. As they go to sleep at night, that's the dark period when the lights are off, their A beta kind of dips down a little bit. In the spinal fluid. But if they're sleep deprived, which is the red line, they actually have higher levels of A beta during that time. So it's suggested in people, just like in mice, sleep deprivation raises amyloid beta levels. This is just one night. And the same thing holds true for phosphorylated tau. So tau kind of does the same thing. So interestingly, they found that they thought this was related to this activity hypothesis. That there's less production of amyloid beta because there's less neuronal activity when you're sleeping. They didn't see an effect on clearance. But there is other data suggesting that during sleep, what might be happening is you're clearing out amyloid beta and tau and other bad things from your brain. And that's based on work from Meka Nadergaard's group at Rochester, which showed in mice that when you inject these tracers into the brain, these red and green tracers, that if you inject them into a sleeping mouse, that they spread through the brain much faster. That's what this picture is showing. So you inject the tracer along it goes along the arteries. These blue things are blood vessels. You can see this green is very close to the artery here in a waking mouse, but in the mouse is asleep, the tracer spreads throughout the brain because there's more fluid flow through the brain in a sleeping animal. And they inject amyloid beta into these brains, and it's cleared out faster when the mice are asleep. That's shown here. And there's some MRI evidence from humans that the same thing can happen, that if you put a tracer into the brain and do MRI before and after sleep deprivation, that the tracer hangs around in the brain longer if the people are sleep deprived. So it suggests that there is a potential clearance effect as well. So there's a production effect of sleep, and there's a clearance effect that both help sleep remove or eliminate amyloid beta and tau and other kind of bad actors in the brain. So this clearance idea has really captured the internet and the lay press. It was in time rinsing of the brain, and deep sleep can remove toxins. You'll see this a lot. And on the internet, there's all kinds of shady websites telling you how to clean your brain and wash your brain and detox your brain and do all these things. None of these probably are true, but so I think that's part of the story. But I think they tend to overlook that there's many other mechanisms probably by which sleep is affecting amyloid pathology and even Alzheimer's disease in general. So one mechanism, Dave Holtzman's group and our group were interested in sort of how do we affect this. And Dave Holtzman's group sort of found that there's this neurotransmitter in the brain called orexin, which is involved in sleep and appetite and metabolism, a bunch of other things. And if you get rid of this particular protein, orexin, from the brain of mice, the mice sleep more. In fact, if you get rid of it altogether, the people that don't have orexin have narcolepsy, so they fall asleep at inopportune times very easily. But you can get rid of most of it, and you don't have narcolepsy, you'll just sleep more. So Holtzman's group back in 2009 gave this drug called elmorexant, which is an inhibitor of orexin signaling. It's a sleep drug. It helps mice sleep. And he found when they sleep, they have to have less plaque. So if you look at the black bars here, those are mice that were given this sleep drug. They sleep a little bit more, about 10% more, and they have a lot less amyloid plaque. Same thing when you delete the gene. If you make a genetic mutant mouse that doesn't have this orexin gene, it sleeps more, about 10% more, and has a lot less amyloid plaques. So this is kind of exciting because there are drugs now, orexin antagonist drugs like suvorexant and lemborexant, daridorexant. There's 3 FDA-approved drugs that are approved for use in insomnia. So these are sleep medications that are used all the time. They're kind of the newest, latest, greatest sleep medications because they're less sedating than a lot of the other drugs. So they're actually better. And in fact, suvorexant is the only drug that's approved for insomnia in Alzheimer's disease. So if you have patients who can't sleep well and have Alzheimer's, you can use this drug because it's less sedating, there's less falls, it's a little safer, but it still works to induce sleep. So we're excited about these drugs as possible preventative agents for Alzheimer's disease. I mean, we can't use them that way yet, but we're investigating that possibility because they're pretty well tolerated drugs that help you sleep and potentially lower A beta. This is some data from my lab with lemborexant, which is one of these drugs that was funded by Eisai, the company that makes lemborexant. You should probably know that. We take mice that get amyloid plaques. We treat them either with vehicles, so control, or with lemborexant at 2 different doses, or with another sleep drug called doxepin, which is an antihistamine, old kind of sleep drug. And they all induce, so this is how much sleep, they induce an increase in sleep. All 3 of the drugs do. It's not a huge amount, but there is an increase in sleep. But the lemborexant in the high dose really lowers plaques, amyloid plaque formation. So you can see that all these little white dots here are plaques in a mouse. Doxepin lowers them a little bit, but not really significantly. But the high dose lemborexant really lowers plaques well. It's pretty striking. So we're trying to understand the mechanisms of that. Like, is that a unique thing to this drug? Is it unique to this type of drug, or is it just a sleep phenomenon in general? I think it's more than just sleep. I think there's a sleep independent component there. And in humans, actually, there's some data. Brendan Lucy, as I mentioned before, he does these catheter studies in humans where they sample CSF. Has some really interesting data that was recently published with suvorexant, which is another one of these orexin antagonist drugs, in which he shows, people that were on placebo or a low dose of suvorexant that doesn't really do much to sleep, they have increases in amyloid beta over the course of the day. But if you give them a dose of suvorexant, just one dose, 20 milligrams suvorexant, that's the green lines here. They have lower A beta for quite a while. Almost looks like about 24 hours. It's gone or lowered. So suggests that potentially in humans, these drugs could lower amyloid beta too. Again, we don't know if it's all through sleep or some other mechanism as well. So we're studying that pretty intensely here at WashU. So I'm going to switch gears a little bit into circadian rhythms, which are kind of intimately connected with sleep, but are a little bit different, and something that my lab works on a lot, and that the Cure Alzheimer's Fund has helped me work on. We all are pretty aware that we have a circadian clock that keeps 24-hour timing, and that's why we go to sleep at night about the same time every day. And kind of most aspects of our bodies have a 24-hour rhythm that's driven by this clock, which is in our brain in this area called the suprachiasmatic nucleus. And the clock is actually entrained by the light-dark cycle. So when you go overseas or change time zones, then all of a sudden the light-dark cycle is off. It takes our internal clock a few days to entrain to that new light-dark cycle. And that's meant to be that way. So this master clock in the SCN kind of drives a lot of other clocks. Almost every cell in our body has a circadian clock that can keep 24-hour time. And they're all synchronized together by this SCN. And one of the main outputs that people are aware of is sleep. Obviously, the sleep-wake cycle is regulated by the circadian clock, or else we would nap all the time and we wouldn't sleep at night. But there's many others. I mean, your gut, your heart, your blood pressure, and your body temperature, and all these things are regulated on a 24-hour cycle. Your immune system, inflammatory regulation, neuronal metabolism in our brain, the blood-brain barrier, synapse function, all these things in the brain have a 24-hour rhythm. So this is what we're sort of interested in. What happens when that rhythm gets disrupted, or how can we kind of target that rhythm as a therapeutic? So we know that people with Alzheimer's disease have not only disrupted sleep, but disrupted daily rhythms of activity, circadian rhythms. You can just measure people's movement, using a little Fitbit type device on their wrist. And then, most people, they move a lot during the day. That's the black marks here. Then they sleep at night. And it's the same day after day after day after day. But these are patients with Alzheimer's disease, probably more advanced Alzheimer's disease, have really a breakdown of their day-night rhythms, almost entire fragmentation of their day-night rhythms. This is a pattern we see very commonly in Alzheimer's, even in the very earliest stages, as I mentioned before, onset of symptoms. It's just not as severe as this until later in the course. So at our place, we did a study to show that people that had biomarker evidence of plaques in their brain, so they get a spinal tap, and we look at tau and A beta in their spinal fluid, and we can tell them, yes, you have plaques, or no, you do not. And the more of this tau to A beta ratio, the higher that is, the more likely it is they have plaques in their brain. And we looked at their circadian rhythms using 2 weeks of actigraphy in about almost 200 patients. You can see there's a nice correlation between having plaques, having more plaque, and having more fragmented circadian rhythms. And this is in people that are cognitively normal. They don't have any symptoms yet. So we can detect these changes in circadian rhythms really early in the disease, potentially could intervene on them, if we think that would be helpful, and to stave off the symptomatic part of the disease. So the circadian clock, unlike sleep is really a behavioral state. It's governed by the coordinated function of lots of different parts of the brain all at once. The circadian clock is really like a molecular machine, like it's a little transcriptional translational feedback loop that's in every cell. So there are specific genes that control our circadian rhythms. If you have mutations in some of these genes, you may have an out of whack circadian rhythm. You may have like a 28-hour rhythm. You might be an extreme night owl or an extreme early bird. Like these things happen. So there's a gene called BMAL1 that I'm going to talk a lot about today. It's kind of the most critical part of the circadian clock. It's a transcription factor that drives circadian rhythms and gene expression. And then there's a bunch of negative feedback loop targets like reverb alpha and beta that I'm going to talk about at some length later today that are not only components of the clock, but they're also involved in a lot of other things like metabolism and inflammation and kind of link the circadian rhythm to those things. So, but one important thing to know is in the circadian clock, if you can delete this gene called BMAL1, it's kind of the linchpin of the whole operation. You will basically break the clock and make the animal or the cell or whatever you're dealing with arrhythmic. So animals that are lacking this gene BMAL1, they go from having very nice rhythms. Even if you put them in dark, they have like a 23-hour period. It's very, very organized activity rhythms to having just complete chaos, like totally fragmented arrhythmic life. They still sleep. They just don't sleep in the same consolidated manner. It's kind of like little naps all day. So you can basically just remember that BMAL knockouts have disrupted, their clock is broken, they're arrhythmic. But it's very handy, like experimental device, because you can knock out BMAL in a given cell type. You can get in a given organ, in the whole animal, whatever you want, using genetics to kind of probe the function of the circadian clock in different ways. And so if we use this method, this genetic method for disrupting the clock, in an Alzheimer's mouse model, an amyloid model, the APPPS1 mice, there's a typo, sorry about that. The mice normally have a nice rhythm in their temperature. We implant little temperature probes and look at their rhythm, and it's completely gone when you knock out BMAL. So these are arrhythmic mice, and they get more plaques. So these little dots are amyloid plaques in a BMAL knockout mouse versus a control APPPS1 mouse, and pretty big increases. So fragmenting the circadian rhythm, it's more than just sleep deprivation. It's actually breaking up this daily rhythm does seem to have detrimental effects and seems to drive amyloid formation in the brain. So there's another way, there's other ways to kind of disrupt the circadian clock. Besides this genetic way, you can use light-induced methods that are non-genetic. We use a jet lag model, circadian desynchrony, in which we just shift the light 6 hours every week. We put mice in a special cage and we can shift the light. This is it simulates eastward travel. So it'd be like flying from New York to London, staying in London for a week. Just when you start to get over your jet lag and get entrained to the London light dark, then you get to fly to, I don't know, the middle of Russia or someplace that's 6 hours further over. And you keep doing that week after week. You kind of once a week, you have to go 6 hours east, 6 hours, 6 time zones east. And when you do this to mice, they still sleep normally. They don't have any stress response necessarily. It doesn't genetically alter any of the clock genes, but it causes their internal clock to always be out of sync with the external environment. So it models things, not only jet lag in our society, but like shift work or, being exposed to light late at night if you're watching a movie at 2 in the morning or something like that. So this is, we think, relevant to experiences that people have in real life. And so if we do this to mice, we take an APPPS1 mouse and we jet lag them for 3 months, it also increases amyloid plaques. So very much like the genetic disruption model, doing this jet lag thing. And you can see when you shift the light over, the yellow is light, it takes the mouse a few days to entrain, to that new rhythm. And that happens over and over again. And we think during that period of time, we don't know the mechanism yet. We're working on that. And actually, Cure Alzheimer's Fund is giving us some funding to help understand those mechanisms. But there's a consistent effect that you knock out genetic clock or you jet lag the mice, it increases amyloid. But that's where the story goes from being nice and simple to being a little more complex and maybe a little more interesting too. So we were saying, well, this is great, amyloid's great, but what about tau? Tau is, really probably worse pathology that causes a lot more degeneration in the brain. We have drugs to get rid of amyloid plaques now. We don't have drugs to get rid of tau. So what happens to tau? Well, interestingly enough, we were very surprised by this, but the exact opposite happens with tau. So when we knock out BMAL and genetically make an arrhythmic mouse in a tau model, this thing called the P3ONS tau model that Virginia Lee's lab made a long time ago, the knockout mice actually have a lot less tau pathology. So this red stuff is aggregated tau in the cortex here, and it's basically gone in the knockout mice. This is just zoomed in. Here's quantification of that. Here's another way of measuring tau. This is highly aggregated forms of tau, the dye called thioflavin S. There's little dots of aggregated tau throughout the brain here. It's basically gone again when we knock out BMAL. So this was very strange to us, but we also know that when we knock out BMAL in a mouse, it causes big changes in the glial cells of the brain. So glia are maybe familiar with them. They're like support cells of the brain. They're a very hot topic in Alzheimer's. I know Cure Alzheimer's Fund is funding a lot of work on glia, but this is kind of our novel angle on glia, particularly astrocytes, which are kind of the main support cells in the brain. They become activated and sort of angry looking when you knock out the clock. You can actually do this if you knock out BMAL in the whole animal, or if you just knock it out in the astrocytes, you get the same phenomenon where the astrocytes take this activated form. And we know that glial cells, including astrocytes, can actually take up tau and degrade it. One of the things they do is, they move around the brain and they take up debris and they take up things like aggregated proteins and they can degrade them, get rid of them. So we wonder if maybe the astrocyte is the key to this. But we next want to say, well, is this just some strange genetic effect of BMAL? So we said, what about our jet lag model? What does that do? So the jet lag, actually, if we do jet lag for 3 months in a regular mouse, it also induces astrocyte activation, just like BMAL knockout does. So you can see this purple is GFAP is a marker of activated astrocytes. It increases pretty dramatically after 3 months of jet lag. So it seems as though there's a commonality there. What about the effects on tau? So we used a different tau model this time, which confuses things a bit, but we think this is a more relevant tau model in some ways, in that you take a mouse that has amyloid plaques, already has them, and then you take tau that you actually isolate it from human brain, from Alzheimer's patients that have passed away. You can take this tau from their brain and inject it into the mouse. And essentially, and Virginia Lee at Penn kind of developed this method. This is like a blob of aggregated tau around an amyloid plaque. You can't see the plaque, it's in the middle there, but the tau aggregates around the plaques, which is kind of what you see in the human brain. It's a little more realistic, we think. So here's another picture of it. This is the orange thing's an amyloid plaque, and the tau is the green blobs around it. So what we did is we let the mice get plaques, we inject them with tau, and then we either jet lag them for 3 months, or we just leave them on regular light dark cycle for 3 months. And very similar to what we saw with BMAL knockout, there's actually less tau aggregation after jet lag. So these are very zoomed in pictures of plaques. The blue things are amyloid plaques in mouse brain. This purple or magenta is aggregated tau, little blobs of tau that you can see there around the plaques. And these little bubbles are generated by the computer to help us quantify how many blobs of tau are around each plaque. But you can see in the jet lag mice, there's still plaques there, but there's less tau aggregated around them. In fact, here's the quantification. There's really a lot, both on the side where we inject and on the opposite side as well. So it has a pretty strong effect on reducing tau pathology somehow, which is totally out of this, not what we expected when we started this project. And actually, this phenomenon, this second round of funding from Cure Alzheimer's Fund, is what we're trying to understand how is this working, and I'll show you some of our progress in that area. How can we figure out how this works so that we can use this to our advantage? So one mechanism that we think may be the case is we noticed that when you knock out BMAL in astrocytes, you get all these activated astrocytes. They express this protein called BAG3, which we were excited about because we know BAG3. It's been shown previously that it's a risk factor gene for multiple degenerative diseases, including Parkinson's. And it's a gene that's involved in the degradation of misfolded proteins. So it's called a chaperone protein. It helps cells kind of take misfolded proteins and degrade them, get rid of them. So the fact that it's highly upregulated in these astrocytes at B-cell knockout was really exciting to us. It's really up a lot. And interestingly, when we look at gene expression in mice that have been exposed to jet lag, we also see BAG3 as one of the upregulated genes. It's considerably higher in jet lag than in control mice. So we did some experiments to look at BAG3 in a little more detail. Could it potentially, be the mechanism by which you prevent some of this pathology? And this is all published now, actually, in this Neuron paper in 2023. But this is looking at alpha-synuclein, which is Parkinson's-related protein. But we have tau data that's very similar. That if we use a viral vector to overexpress BAG3, that we get less alpha-synuclein aggregation after we inject alpha-synuclein into the brain. So under normal, if we use a control virus that just doesn't express BAG3, there's quite a bit of alpha-synuclein, these little red dots that aggregates in this area of the cortex. But if we overexpress BAG3 in this area, there's a lot less alpha-synuclein. So we think BAG3 is protective against tau and alpha-synuclein. So that leads us back to the clock. So how does it induce BAG3? Like how are these things connected? And the answer is through this gene called reverb alpha, which is the one that my first 2018 Cure Alzheimer's Fund grant was to study. At that time, we had no idea about this pathway, but we subsequently kind of worked on this. So reverb alpha is downstream of BMAL, so it's part of the clock. But as I mentioned, it's involved in a lot of other things. You can see it has this very nice circadian rhythm in the brain, and when you knock out BMAL, you kind of lose reverb alpha. But importantly, when you knock out reverb alpha in the brain, you get really high levels of BAG3. So we think that BMAL signals through reverb alpha to control BAG3 in astrocytes. So who cares about reverb alpha? Well, the reason why we're interested in reverb alpha is because it's circadian. We know it's involved in the liver, in metabolism and inflammation, and there's a lot of data on it in other organs, but it's not really studied in the brain. But most importantly, it's a receptor. So these other things are transcription factors. You can't really make drugs against them very well. But reverb alpha is a nuclear receptor. So there are drugs, agonists and antagonists, inhibitors and activators that target this protein. So we could potentially use these drugs to kind of manipulate the circadian system. And you can do that because it's not a key part of the circadian clock. You can actually do that without completely messing up the circadian clock in general. So we think this is an area where we can potentially pharmacologically intervene to sort of get the clock to do what we want it to do. So according to our theory, if we knock out reverb alpha, reverb proteins, we should have a protective effect in these tau models. And we've now done that in part with funding from Cure Alzheimer's Fund. We've made mice that have, these are tau mice, these are P3ONS, PS19 tau mice, the same ones I showed before, that have this reverb alpha gene knocked out postnatally in the whole animal. And you can see these little dark dots are tau-bearing neurons, and you can see there's less of them in the reverb knockout brain. There's also this red stuff is MC1, it's misfolded tau. There's a lot of it in the control mice, but in the mice lacking reverb alpha, there's a lot less of it. And it actually prevents neurodegeneration as well. So that's exciting. I mean, that kind of fits with our story. These mice, I don't show it here, but they have high levels of BAG3 as well. And so, as I mentioned, reverb alpha is a druggable target. And so we have obtained this drug called SR8278. One of our collaborators here at WashU actually is like the world leader in reverb alpha drugs, which just seems like a very unlikely but very helpful coincidence. They provided us with this drug, and we gave it to some tau mice for a month. And you can see this brown staining is aggregated tau pathology. The mice treated with the drug have less, and so you can see that's quantified here. And this is just two different ways of quantifying the amount of tau. And there's less of this kind of pathological inflammation in the brain as well. So we're excited about this as a potential path forward to kind of leverage some of these basic science findings into therapeutic possibilities in a kind of a novel pathway to help prevent AD. So we're still at the beginnings. We have the tip of the iceberg here. We don't understand the function of all these genes. We don't understand how exactly. We think BAG3 is involved, but I'm sure there's a lot of other stuff going on. In fact, we already know there's other things, by which sleep and the circadian clock regulate A beta and tau and neurodegeneration and glial function. So I think we have an exciting start. We have at least a first a step forward with this reverb alpha BAG3 axis that we think we can target. But there's a lot more to be learned, and certainly organizations like Cure Alzheimer's Fund are making that happen.